Hello, my name is Bernard, and I'm delighted to provide this short demo about tissue knowledge management on behalf of the Open Physiology team. Part of this demo makes use of data imported from the Human Protein Atlas. The motivation for the Open Physiology effort is to articulate the relationship between tissue function and molecular function. We will achieve this articulation through the representation of and inferencing over physiological routes across the body using our ApiNATOMY toolkit. A key benefit of this work is that it supports the formal description of disease mechanisms in terms of an interference over these routes. In practice, physiological routes consist of a concatenation of anatomical structures that are key to regulating when and where molecules are allowed to co-locate and interact, interaction being essential to molecular function. In order to describe these routes, we generate tissue parcellations from three-dimensional histology images known as primary functional tissue units, or PFTUs. Geometrically, a PFTU consists of a cylindrical diffusive field with an 80 micron diameter and 60 micron length. The long axis of this field is aligned on a central advective conduit, such as an epithelial or endothelial vessel. The central vessels are connected to ontology-based topological representations of routes of long-range advection. From a mechanistic perspective, therefore, PFTUs represent a point of transition from advection-driven processes to diffusion-driven processes. In particular, we are investigating the role of the PFTU as a tissue domain that acts as a scaffold for the tissue-specific and molecule-specific data about permeability and flow relevant to the regulation of molecular interaction, such as membrane permeability of the cellular constituents of a PFTU, permeability of the central conduit wall, as well as the parenchymal cuff around it, flow of the central advective conduit, and the degree of overlap between the diffusive fields of neighboring PFTUs as a result of processes such as development, growth, as well as physiological or pathological adaptation. In that sense, ensuring semantic interoperability with Human Protein Atlas holdings is a critical step in articulating data that is both tissue-specific and molecule-specific. We show here our initial steps in overlaying both immunohistochemistry images and the interpro architecture of proteins for which antibody stains are shown in these images. We embed this data in the precise context of our physiology circuit board, and this interoperability has been achieved in terms of a common standard for uniform resource IDs for organs and proteins. In this view, we have filtered the Human Protein Atlas for protein pairs that are involved in long-distance interaction. We see here, for instance, lipoprotein A, secreted by the liver, and its amyloid beta receptor expressed in the colon. In ApiNATOMY, such circuit boards can be automatically constructed to map out the route required for these ligand-receptor pairs to meet, taking into account those permeabilities and flows that are relevant to these long-distance interactions. The calculation of such routes are of interest, of course, to the study of processes of absorption, distribution, metabolism, and excretion relevant to the handling of drug by the body. The next step is to advance the coverage of this semantic interoperability to bridge cellular annotation in the Human Protein Atlas to cellular annotation within PFTUs linked to the physiology circuit board. Thank you.